going to talk about this project of queen fertility testing that we've been doing this year at Tech Transfer. So just to preface this, this is the first time we're presenting these results. So there's still some work that needs to be done, and I'm sure I'll give an update. We will later in the year or next year as well. So this is the beginnings of this project. So I'll take you through it, what happened this year. So the reason that we started this project in the first place was due to the ongoing concern that beekeepers have identified some serious problems with queens in recent years. Just poorer queens in general. So whether that's early supersedures or more frequent supersedures or more drone-laying queens, it's been identified by beekeepers in Ontario that this is a big concern, and it's something that Tech Transfer should look into. So that was the whole basis behind wanting to get this kind of setup started.
Are you able to turn the volume up a little bit?
Yep, on speak a little louder. Sorry, if you can't hear me at the back, I'll try and speak up. So the goals and the scope of this project, I'll give you an idea of. First of all, we just wanted to develop a protocol for being able to test queens, the fertility, and specifically with this project, we're looking at the viability of the sperm that's stored in the queen's spermatheca. So we wanted to get into the lab and be able to do this with a consistent success. And that's something that I think we've done. I'll take you through all the methods and the protocols that we use, but I think that part of this has been a success. We've been able to do that this year. And maybe to identify the mechanisms behind the problems with these queens. So, like I said at the beginning, there's been problems identified, so we want to just try and figure out why those problems are happening and maybe if there's a way we can fix them. So the kind of end goals of this project were to get this information into the beekeepers' hands and hopefully allow them to be able to test for this thing regularly, be able to select for increased fertility. So maybe this is something that can be passed on through generations of queens, and if you're selecting for queens that are more fertile or show better viability of sperm, then their offspring will have those traits as well. Another part of this project that I thought would be pretty useful is to be able to help beekeepers with their management styles. So maybe testing for sperm counts. If you're getting low sperm counts, for example, you might want to change what's going on in your mating yards. So maybe you don't have enough drone source colonies, maybe the area that you're mating your queens in isn't good for them, or maybe if you have a few areas that you're mating queens in, you can test queens from those different areas and find out what's working better. So those are just some possibilities that hopefully beekeepers in the future are going to be taking advantage of. I'll get right into the methodology of this project. It starts with getting queens from beekeepers. So we did this project with the involvement of the Ontario Bee Breeders Association. So people that are have been involved in the past with the Tech Transfer breeding program. Also, other beekeepers that weren't involved with that program contributed some queens this year, and we're very thankful for that too. So it starts here with the dissection of a queen when she's alive. So we dissect out the spermatheca, and we do it while the queen is alive in order to best preserve that sperm. So you can imagine if you were to dunk the queen in alcohol and kill her before, you're going to damage the spermatheca most likely and some of the sperm that goes along with that. So that's why it's important to do it while the queen was alive. There's potential that we could have anesthetized the queens with carbon dioxide is a common method of putting the queens to sleep, but it wasn't really necessary. It would have been a lot of extra work and expense. So this is commonly done, a live queen dissection. So what you end up with is the dissected spermatheca. So that little white ball on the end of those forceps is the spermatheca out of a queen. There's a few things you can tell right off the bat about the spermatheca. First of all, a virgin queen that has not been mated at all has a pretty perfectly clear, looks like a little crystal ball spermatheca. So this one, you can see it's white, and due to the photo quality, you can't quite see all the detail, but it has kind of a marbled, creamy white appearance to it, and that's a sign of a well-mated queen. These are some of the other things. This is a typical what the lab bench looks like when we're doing this. The tissue grinders are these things over here that look just like test tubes, but it's a really specific device that's finely calibrated. So we dilute one spermatheca in 1 milliliter of solution, and we grind it in the tissue grinder. So what it does is it ruptures the organ, but it doesn't destroy the cells. So that's the idea of the product. That's why it's such a fine piece of equipment. Then the other thing here that I've got on is Kiev solution. So when I said we dilute one spermatheca in solution, we went with Kiev solution on this, and that's from a big literature review that I've done, as well as some experimental testing. So I'll get to the results later, but we were seeing quite low viability of the sperm. So we thought at first there could be a problem with our protocol, with the methods that we're using, and one of the things would be the solution that we're diluting the sperm into. If it's the wrong concentration, or there's the wrong materials in there, or the wrong osmolarity of the solution, then we could actually be killing sperm cells in that solution. So I actually went through 4 different solutions in the lab and saw no differences between any of them. And these were all solutions that had been tested throughout experiments earlier. And they really showed no difference. So Kiev solution was relatively simple to make. All the ingredients were in the lab already. So that's the solution that we went with on this. And then the samples are dyed. So you can see at the front of the screen there, I'm using a micropipette to move parts of that sample that's been crushed in solution. And then we add to that fluorescent dyes, which I'll show you a bit of in a minute. And that's another part of the methods that we toyed with a lot. In particular, the one dye is a bit more sensitive than the others, so we played with the concentrations of these dyes as well as the incubation times of the samples. So we settled on something that worked best for us. So there was a lot of trial and error involved originally with this project. This is the microscope. So I haven't mentioned it yet, but this is actually in Dr. Ernesto Guzman's lab at the University of Guelph. So this is where we've been doing all this fertility testing throughout the year. And I have to give a huge thank you to him and Hamid at the lab. Have been really great in letting me come in, sometimes on very short notice, to get these samples done. But this is the microscope that we use. So on the left of the picture there is the light source that is used for the fluorescence part of it. The microscope can also be used, it's just a regular light microscope as well. So we use it for the regular sperm counts with the hemocytometer, which I'll show you in a minute too. So this microscope, Ernesto's lab got this a few years ago with actually some money that was contributed from the OBA. So this is part of the money that you all have put forward in the past. We're still making good use of it in Ernesto's lab. So it's getting a lot of use now. Okay, this is a hemocytometer, and I'll apologize for the picture quality. You can't exactly see, it doesn't look as good as when I see it firsthand under the microscope, but it does kind of show you how difficult this can be. So seeing honeybee sperm, they have really long tails compared to their heads. So at times when you're seeing a lot of them, it looks like kind of a mess under a hemocytometer. So it's really, it can be quite time consuming to figure out just how many sperm cells are on the grid lines. So the box that I've outlined there is an example of one area that we would count the number of sperm cells in, and we'd actually do 5 of those on one side and then 5 again. So that gives you, you do a calculation from that, and that'll tell you how many cells of sperm are in that sample. So while I'm doing this, I've been incubating the samples with the dye. So this is what they look like. Again, I got to apologize a bit for the picture quality. It was really difficult to take some pictures down this microscope, especially in the dark as we do it. So these are the dyes on the left, the Cyber 14, and I actually switched partway through the year to this other dye. It's called Cyto 16, but they do the exact same thing. And on the right, the dye is called propidium iodide. So we add both of these dyes to a sample of sperm, and we let it incubate. And with our methods this year, that was about 25 to 30 minutes at room temperature that they incubate. And then the way it works is dead cells look red, and living cells look green. So just to point out, these two pictures would actually be one field of view. So I haven't moved anything on the microscope slide. I've just changed the filter. So when you're looking down through the eyepieces of the microscope, you can change the filter on this fluorescence microscope, and it just allows different wavelengths of light to get to your eye. So to look at the red cells, we use one filter, and to look at the green living cells, we use another. So in that same field of view, I'll just switch back and forth that filter, and you can see. So in green, the green dye is the one that's a little more finicky, but these I'm pointing out there, and there's a bunch there. There's another one. Those are the nucleus of the sperm cells, and the red ones are pretty obvious. It's a bit overexposed. It doesn't look quite that bright, the red especially when you actually look at it, but from the picture taken, it looks a bit too bright. But the red was really easy to see all the time. That dye worked really well. The green was the one we had to play with the concentration with quite a bit to be able to make sure we were, first of all, staining all the cells to make sure that they were well stained, and then make it easy to count them too, so have them bright enough that they were all being accounted for. Just a quick note, the way these dyes actually work, the propidium iodide will only penetrate dead cells because there's, on the cell membrane, there would be an active pump excluding that molecule. So when a cell is dead, the red stain is able to go in, and both of these stains bind to the DNA in the nucleus of the cells. This is a slightly closer shot of the green, the living cells. You can see here, there's these curls that are just really faint, but those are the tails of the sperm cells. So on a really good sample, when it's dyed just right and it's incubated really well, you can actually see the tails glowing a little bit. And I don't know for sure, but I think it's almost like that tail is acting like a little fiber optic cord. So when that nucleus of the cell is stained really bright, you can see the tails going. And a lot of times we'll see them moving around too. The living cells are often pretty motile under the microscope still. Okay, so getting into some of the results here. This is just the sperm count, so the number of sperm stored in a queen spermatheca. And I've divided this year's results by month. So first of all, I only included on this graph, it only includes queens where I knew from the beekeeper when those queens were mated. So there were some queens that we received where I wasn't sure when they were mated, or some of them were even mated from the previous year. So I didn't include those. I wasn't trying to guess at when they were mated. So this shows you over time the sperm counts. And you can tell June and July we got a lot more queens to sample than queens that were mated in August. That's just normal for the industry here. So if anything, there's a slight upward trend. I just added this line just to kind of illustrate to you guys. You'll see in the averages there is a slight increase towards the end of the season. I'm not sure. I don't think it's a really significant trend, but still, there's something that's there. And you can see on the left side millions of sperm stored. So on the next slide here, I've got the averages. Those are the numbers that the averages of all those dots on the graph by month, and then the total for 2012 here. That's per queen. On average, 4.4 million sperm stored in her spermatheca. You can see in August there was a big spike, but that was a pretty small sample size too. I think only 4 queens I knew for a fact were mated in August that I included in that part. So you'll see on the right side the numbers of queens included in those totals. And so everything from the individual months of 2012 don't quite add up to the 67 queens we were able to sample, just because some of those queens I wasn't sure of the date. But regardless, all those queens are included in the total 2012 average. And then this is something that Tech Transfer has actually been doing for a lot of years, since well before I was around. So we have queen quality survey results back from 2002. And all I've listed here is the average number of sperm stored in a queen and how many queens were submitted in those years. So a couple noteworthy ones, 2008 and 2006 there look quite low. Yeah. You'll notice that the sample size is a bit smaller, but also part of the reason that we have been doing this queen quality survey in the past was to check for problems in queens. So a lot of the queens that were submitted in the past, beekeepers had identified problems. This queen wasn't laying properly. I'll send her into tech transfer and see how well she's mated. And so I think that needs to be factored in. With a lot of these averages, they're going to appear too low, and that's probably because they are. A lot of those queens weren't performing very well, and they were factored into those numbers. But still, back in 2002 and 2003, the averages were really respectable. And I was amazed. In 2002, all the participation, 54 queens were sent in. So that was really encouraging to see. Now we get into the viability of these same queens. You can see a much stronger trend here from month to month. But look specifically at the y-axis there, the percent of living sperm that's stored. The average, which I'll show you on the next slide, is going to be just over 50% for this year. And we had only a handful of queens that were over 70%. So that's the big concern that we've got with this project right now, is what's going on with this. Okay, I'll show you. Here's the averages broken down by month, and then the total, 51.3% this year. Okay, so there's a couple notes to make with the queens that we actually got in. Just some little kind of anecdotal things that were noticed with the queens that were surveyed. So I was able to test one imported queen from the United States this year. I know one queen, you really can't make any conclusions about it, but that one queen in particular had a really low sperm count. It was less than 2 million sperm that was stored, and the viability of her was below average. I think it was just over 40%, but not wildly out of our range that we're seeing in Ontario. Also to note, I received 4 queens from a couple different bee breeders that were that were mated specifically in an isolated area, and it happened to be also an area with some different agricultural practices. It was just different in a lot of ways. And those queens actually tested well above the averages, and especially in the viability. So when I said the total average was about 50%, those 4 queens would have averaged about 70%. They were like all among the top queens in the test. So there could be something with that, and I'm going to talk to those breeders in particular about getting some more information about that. We were also able to test some supersedure queens that were given by a breeder. So most of the queens that we get in are raised intentionally. So they're hopefully using good techniques with their cell builders, and they're trying to raise good quality queens. A few supersedure queens were pulled out of hives, and I was able to test those. The viability of those queens was about average, maybe a bit below average, but again, not out of the ordinary. But the sperm counts of those queens were significantly lower than the averages. So, that could just be the area that they were mated in. They were just in production hives. So, but I think it's more likely that the quality of those queens just wasn't the same as when we raise our own queens. So they're under a bit more pressure to put a queen out more quickly, and those queens weren't storing as much sperm. So with all that being said, the last thing I want actually is to turn this into a competition or a fight between breeders or between imported queens and our own queens. I think there's just a lot to learn from all this, and if one person or one area is doing something better than someone else, then I think we just need to learn from that and not try and turn this into a competition like these 2 queens. So for the rest of this, I want to try and just put all this into context. So that 51% average viability, I said it was low and it was of concern, but I want to introduce you to a few other studies that were done and how we can try and make sense of it. So I think this study here by David Tarpey from North Carolina State University is really the best study of its kind, and really one of the only ones that I'm comfortable comparing our results from this year to. So he was able to test 59 queens, and these were all from queen producers in California, actually. So I'll give you a quick comparison of the numbers. Our sperm counts were about 4.5 million, and his were right about the same. It's the viability that was the big difference there. So our average was just over 50, and his average was just over 80% in those queens. I think it's important to point out the range of the samples tested. So the lowest viability that we were really seeing was just over 30%, and we saw a few over 70%. Their range they had some low numbers in the range too, as low as 20%. But the best queens in their study were approaching perfect. Like almost all of the sperm in the spermatheca was alive. So it's kind of discouraging that we didn't have any queens in Ontario where we saw reaching that really high number, but it's also encouraging that they did see some low numbers the same as we saw this year as well. So we had, our range was tighter. They were spread more all over the place, but they did have some low queens as well. So, maybe that's to say that it's not completely abnormal that we're seeing some lower sperm or some lower viabilities. Okay. And then some other work that's been done here in Ontario was by Meghan Taylor. So Meghan actually helped us at Tech Transfer put together this protocol. She was in the lab with us in Ernesto's lab last winter for a couple of days, helping get things sorted out because she had some experience with this kind of work in the past. But her focus had been cryopreservation. So she was collecting semen from drones and freezing it, trying to keep it over long periods, and then thawing it and testing the viability then. So that's why she knew a bit about viability, but she also knew quite a bit about drone semen quality. So her study, this is from the results were published 2009. She looked at a few different strains of bees. They were actually from a few different bee breeders in Ontario. But these are the viability percents of fresh drone sperm that she collected in the field. And you'll see it's mid 60%. So there's some similar work going on in Quebec right now. In Pierre Giovinazzo's lab, who's at our spring meeting last year, he's doing some great work with queen quality in Quebec. So he has a grad student right now that's doing a pretty similar survey to what we're doing, what we had done this year, looking at it at different times of year, but she's specifically just looking at drone quality. So I'm not going to present all of her results because I don't have them all, and she's still working on this project. But speaking to her at Apimondia in Quebec this year, the average viability of all their drone sperm was 64% this year. So it's right about in line with what was found in Ontario a few years ago. So that's important to note. So here's a graph. This is by Anita Collins. So Anita Collins was a researcher with the USDA. She's retired now. But she did a lot of work on cryopreservation, like what Meghan was doing, with artificial insemination of queens, and a lot of work with the kinds of things that we were interested in here. So this graph, it needs a bit of explaining, so I'll do my best. So you can see the dark bars are the viability levels of the sperm that she inseminated queens with. Along the bottom was a mix. These were her mix ratios. So, for example, the one that says 25, it was 25% fresh semen out of a drone and 75% semen that she had killed intentionally and mixed those together. So those ratios go up from there. And then she inseminated the queens with these ratios of sperm, let them go in nukes and begin laying. She measured their brood quality, and then later she pulled out those queens, dissected them as we were doing, and checked the viability in the spermatheca. And some really interesting things are noted here. I'll start in the middle of the graph with the 50, 75, and 100% mixtures. What it actually turned out as, for the 50% mixture, for example, it was 50% fresh semen, and the average viability of that turned out to be just over 40%, as you can see on that dark bar. And for the 75% mix, the viability was about 55% in that. So you inseminate the queens, say with that 55% viable semen, dissect them later, and you find about 55% viable sperm in that spermatheca. When you get into the low range there, with just a 25% living sperm inseminated, you see a big increase. So the way it works, when a queen is inseminated, when she mates with the drones or whatever, that sperm has to migrate to her oviducts and into the spermatheca. So only living sperm can actively migrate, obviously. But there's what Anita Collins found anyway was that there's a real flow that's created with that living sperm, and apparently from those ratios in the middle, it carries along a lot of dead sperm with it. With that first ratio, it didn't carry a lot of dead sperm, I suppose because there was so much of it, there wasn't that same kind of flow created. But the other interesting thing to note was when she examined the brood quality of these queens, the ones in the middle with the 50, 75, and 100% fresh semen ratios were laying completely normally. Even though they had only stored between 55 and 65% viable sperm in their spermatheca, they appeared completely healthy. They were laying good quality brood patterns. There wasn't excessive drone brood. They weren't drone layers or anything like that. And that's what we're seeing in our queens in Ontario. We're seeing, a viability around 50%, but these queens are still productive, and they appear to be a good quality. The difference might be in their longevity. Also to note, in that 25% ratio, it looks really good that there's a lot of viable sperm stored, but there wasn't a lot of viable sperm stored. Most of it was viable, but there was very little sperm stored in their spermathecas, so those were the queens that were actually laying very poorly. Some of them became drone layers quickly. Some of them didn't lay at all, actually. So I think that puts a little bit of context into what we're seeing in Ontario. So, there's lots of questions about where do we go now. I think the most important thing is the last point on this slide. We need another year of data. We need to do it again and ask some more questions, maybe get some more groups of queens from different areas or that are treated differently. We just need more data to try and understand what's going on. There's potential for collaboration with Quebec. I mentioned Pierre Giovinazzo, and he's really interested in, at the very least, getting his lab to test some drones from Ontario, or who knows, we might share some of the results of our own studies with each other and try and put something really good together. Like I said before, with the materials and methods of this project, there was a lot of trial and error in trying to make everything work the best that it could. But I'm still open to trying new things in the lab, that's for sure. You can't just, you can't sit on your hands when you think you've got something that's going well. So, I'm open to trying new techniques or doing things maybe a little bit differently if anyone thinks it would make a difference. And I mentioned that one imported queen that we were able to test before. I'd really like to be able to test more. The thing with this funding is it's part of a project where we're trying to help the bee breeders of Ontario. But if we can test the quality of queens that are coming into the province and try and establish some kind of some baseline or quality control, even something that'll help the industry with the queens that we're getting in Ontario, I think that'll benefit everyone. One thing I'd like to do is send out a management survey and find out the types of queen rearing techniques that are going on, more about the locations of mating, the timings of matings, and really get a better picture of what these queens went through before I got them in the lab and tested them. So I'm not sure how much time I have left, but 10 okay, good. A bit of discussion here. This is regarding I wrote an article for the Ontario Bee Journal that discussed some of the results that we were finding and some possible solutions is what I was really trying to get at. So the big focus of that article was varroa and miticides during drone rearing. So I think it's really important to focus on the way you're raising your drones. And this point I put up there is really important. Even under optimal conditions, when drones are raised in pristine hives, still less than half of them are surviving to sexual maturity and mating with queens. And that's normal. That's what researchers all over have found. When you add some chemicals to your hive, like a chemical treatment or even organic treatments like formic acid at the same time as drone rearing, you decrease those drones by down to about 40%. But when you add varroa mites to the equation, if your drone source colonies that are producing drones are infested with varroa mites, you're getting less than a quarter of the drones that were originally laid by that queen surviving to go out and mate. So it's so important, like with everything we say, every part of beekeeping now, it's incredibly important to manage varroa properly. And it's important to manage the treatments that you're using too. So that was, all those results are kind of a review of some American Bee Journal articles that were written back in '99. Another thing, along with proper drone rearing, is proper queen rearing. So I mentioned those supersedure queens. Those were just an example of some queens that were not reared under optimal conditions. And so, it's not very likely that Varroa are going to affect your queens during their development. And you're probably not going to put some chemical strips in your cell builder, but maybe there's residues in your wax, or maybe the colonies that you're using for your cell builder were treated with chemicals right before, in that spring, before you made them into a cell builder. So there's potential there that those types of things, management, could be affecting the queen's development. There's no reason that they wouldn't be, really. And so, just apply the same principles you use with trying to rear healthy drones to raising healthy queens. And, the proper cell builders, having strong cell builders and used in the right way is really important to getting the best quality queens you can. And I think I'd be missing something if I didn't mention some issues with agricultural pesticides. Not only what we've seen in Ontario this year, but the fact that that pesticides are in our environment and persistent there is something that we need to pay attention to. So I know there were beekeepers this year that had some serious problems trying to raise queens after they had incidents with acute pesticide poisoning. And lots of talk about drones too. I mean, I saw firsthand some of the things that go on with drones that were affected in those colonies. Basically, drones dying in some strange ways. So there's no doubt that those things are going to be affecting the health of your colonies with regards to raising drones and trying to produce queens too. And we can think all day about some things we might be doing wrong with our hives, but every little bit counts when you're trying to manage your bees. Doing the best job you can ultimately adds up to produce your results. So that's the end. I really wanted to thank this project. It was a big undertaking, and all the people that submitted queens this year, they were donated to us basically. And the bee breeders got back some valuable information out of it, I'm sure, but they were just giving us queens. And it wasn't only those 67 queens that I mentioned throughout the survey, but we received queens in the winter from some other bee breeders to practice, to get this protocol down. And so I have to thank those people who donated queens, because that made this happen. And Dr. Ernesto Guzman's lab, Dr. Guzman himself, and everyone there that I've listed. When I stain the samples with these dyes, I have to do it in the dark. And the fluorescence microscope only works in the dark. So the dyes are light sensitive, they can't be exposed to light. And when I go in there, I'm constantly turning on and off the lights. And so the patience that everyone there has displayed has really impressed me. So I've got to thank you all for that. And Ernesto always says that sometimes he sticks his head in because he thinks everyone's gone home, but the lights are just out and I'm doing work in there. And then Meghan Taylor came up from Washington State University, where she's doing a PhD right now, and helped us in that lab try and figure out the protocols in the first place. And so I have to, a big thanks to her. And so the funding for this project is through the Agricultural Adaptation Council, and the program that it's under is the Canadian Agricultural Adaptation Program. We just call it CAP. So that's it. Thank you.